So when you look at this equation, it's actually capturing both what's happening for the light reactions as well as the biochemical, the reduction of the CO2 or the fixing of the CO2 into sugars. And when we look at the light reactions, we're going to see that water is actually the initial electron donor that kind of kicks off everything. And then for part two, that's when we start talking about CO2 coming in through the stomata and getting fixed into carbohydrate. So this is a very big picture idea equation. But when you just zoom in on the light reactions and you look at it more carefully, that's the goal for the plant. The plant wants to make plenty of ATP and NADPH. And so this process that has evolved is designed to allow for that to happen. And the reason they want to make all of that is that it's then used to make their sugars. So definitely the light reactions and the biochemical reactions that take the CO2 and fix it into sugars, the carbon fixation, they're linked together. So when we think about what's happening in the light reactions, we need to think a little bit about free energy, which you've learned about in intro chemistry classes. And so delta G is your free energy, and this is the equation for Gibbs free energy. That's the energy available to do work. And if you're curious, those are all the definitions to jog your memory. We're not going to work through these or calculate free energy, but understanding the idea of free energy as a whole is important, because when you look at these reactions, you can determine if it's going to happen spontaneously or if it needs an input of energy. And so if you end up solving for that equation and your delta G is positive, that means that particular reaction where they calculated the thermodynamics, it's going to need an input of energy. If the fancy word for that, by the way, is endergonic. I don't care that you know that. I care that you understand a positive delta G, you got to push it with energy. If your delta G is negative, then it should happen spontaneously. The reaction should happen spontaneously given the right set of ingredients and all of that, it's just going to happen. So exergonic. So when you look at the overall overall photosynthesis equation, going back to that, so if you calculate the thermodynamics to get CO2 fixed into or reduced into sugars, that's definitely a positive delta G. That just doesn't spontaneously happen. And if you think about it, like right now in our atmosphere, there's CO2. Right now in our atmosphere, there's water. And yet we can't just stick out our tongue and have sugar magically show up, right? It's not like we can do that. So if it's raining or snowing out, it might be fun to catch snowflakes on your tongue, but it's not going to feed you sugar. So it's just not happening spontaneously, unfortunately. So you have to provide quite a bit of energy in order to get that to happen. On the other hand, if you take this equation and put it in reverse, switch the arrow, and think about breaking down carbohydrate into CO2 and water, that would be the idea of oxidizing. In this case, we typically think about glucose. If you break down glucose, that's a negative free energy, which you're doing right now. If you had something to eat, your body is doing that right now. So glycolysis is an example of that's overall, it's a negative free energy. So when we talk about oxidation and reduction, we're talking about redox reactions. And that's the idea where we're going to see electrons are getting transferred between different molecules. And for the light reactions, it's a long series or chain of molecules that will be passing along the electrons from donor to acceptor. This is often how they describe it, where they give you some made up molecule, A, H2, and then they say, well, if you dissect that molecule, when you think about H2 in particular, Each hydrogen atom is composed of one proton and one electron. And so this is the way to depict that. When you pick this molecule apart, H2, you end up with 2 electrons, 2 protons. So what they're showing here, when you have molecules interacting, so here in this bullet, you have A and B, AH2 and B are going to interact. And you see that there's a transfer here. A donates off its H2 to B. So when you look at that reaction, it's not like you see an electron getting transferred per se, but you have to remember that each hydrogen has a proton and an electron. So you are transferring electrons when you transfer that hydrogen or the H2, hydrogen gas. So very, very often when you look at these reactions, you'll see hydrogen getting transferred from molecule to molecule. But just remember there's an electron coming with it, right? And I don't know, but for some reason, when that got explained to me in that way, that made a whole lot more sense to me when I was looking at this idea of redox reactions. Some terminology that you'll hear when they are looking at redox reactions, they'll say, oh, that molecule is a strong reductant. What that means is that it's really good at giving up electrons. Chemically speaking, it's favored to give up electrons. So we're going to find out that ferredoxin, FD, I'll talk more about that one, is actually pretty good at this. It's pretty good at giving up electrons. On the other hand, if you're a strong oxidant, that means you're really good at accepting electrons. By definition, that also means you don't want to give up electrons. You're good at accepting, but you don't give them up. Not without a fight, so to speak. So an excellent example of a strong oxidant is water. Water is good at accepting, really terrible at donating. It's pretty stingy or selfish. It wants to hang on to those electrons. And in order to convince water to give up an electron, you have to supply a lot of energy to get that to happen. They often depict these ideas like this, where you're looking at the arrows depicting electrons. You have these paired electrons here in the ground state. And then they say, if you think about the chlorophyll, they're trying to talk about chlorophyll now in particular, because that's what we're zooming in on with the light reactions. We're going to see how a particular chlorophyll molecule in the reaction pathway gets excited by that light energy, and that will get one of the electrons into that excited, hopefully triplet state, ready for it to get passed off to an accepting molecule somewhere. So this is often how they show this idea. And then once that electron gets passed off to another molecule, that will have been a redox reaction that occurred, okay? Now, here, they're trying to show this idea that we already looked at last time, where you have these antenna pigments that are surrounding the so-called reaction center. So that's what they're depicting here. So all the lighter green are the antenna pigments, and they're transferring that energy to the reaction center. And that absorption then of that energy by that chlorophyll in the P680, that gets that chlorophyll electron excited into the excited state. So when you see it written like this, P680 asterisk, when you see that, that means it's excited. And that electron's excited and it's ready to be given off. And then they're trying to show here that you get this flow of electrons through a chain process, the electron transport chain process. And you see there's another photosystem involved here. And in this diagram, what they're kind of emphasizing is that you have these two photosystems photosystem 2 on the left here, photosystem 1. So they're players in the game of this electron transport chain, and that's where we see the light energy really having a strong impact. Once that light energy gets to the photosystems, it's capable of getting that chlorophyll electron excited and then hopefully donate it off to the correct acceptor. And you can keep this process running. They've also put it here, on this scale on the y-axis. They call it the redox potential. So when it's down low toward the bottom, when it's down here, that means it's pretty much in the unexcited state. And you really have to hit it with some energy to get it excited and up into a more excited state. So oxidizing, what they're saying there is that it doesn't like to give up electrons. It can accept electrons, but doesn't like to give up. And then reducing up top here, that means once it's in that excited state, this chlorophyll that got excited by that light energy, now that electron is in a more of a reducing state where it's like, yeah, I can give off that electron. I have a sufficient energy to do that. So when they start looking at the light reactions, they can map it out like this on this energy scale. So this energy depiction, this energy scale depiction is called the Z-scheme, because if you turn your head sideways, this represents a Z pattern, the way it is. But basically what they're showing here is that you have, again, this EMV or EM measured in volts. That's your energy. All right. And it goes up as you go up the y-axis. So the way to interpret this is that you can see a negative scale here up top. So you have that 0 point and in the middle, then you see negative 0.5, -1, -1.5. So when you get into the redox potential, this is the EM is called redox potential. When you get a negative, that means it's in a state where it's very capable to donate off electrons, give up electrons. If you're in the positive end of the scale, that means those molecules found in that region, they're good at accepting electrons, but they're not good at donating. So the Z-scheme is trying to show that when you give light energy, when those special chlorophyll molecules in the reaction centers get hit with that light energy and get their electron excited, that pushes them up the tower, so to speak, into a negative reduction potential. That's good, because now it's like, okay, now I can donate off that electron. I have enough energy to do that. And then here, all these different letters and acronyms, those are the different players in the game. Those are different molecules that will take that electron, pass it on, keep on doing that on down the line. You see here the other photosystem coming into play, also getting hit with light energy. And it's special chlorophyll, one of the electrons getting excited. And then you see more players here in the game, taking those electrons and passing it on down the line or down the chain. Until you see at the very, very end, NADP plus. So we call NADP plus the ultimate or final electron acceptor in this whole process. And when it accepts an electron, then it becomes reduced to NADPH. So that's a huge goal of the light reactions. And then if you go all the way back to the very beginning with photosystem 2, one of the things they're trying to imply here is that you have this very special oxygen evolving complex, they call it. It's part of the photosystem 2, but it's a very special region of photosystem 2 that allows some of that energy from being absorbed by sunlight energy, allows for the splitting or stripping off of electrons off of water. So water is very low on the reduction potential. It's got a positive value that's trying to say it doesn't want to give up electrons. You have to hit it with energy. So sunlight energy funneled into photosystem 2 like that allows for water, electrons to be taken off of water. And that's what they're trying to show there. So this black arrow coming off the oxygen evolving complex, that YZ, they're trying to show the black arrow is electron flow. So they're saying, yep, we got electrons stripped off of water. It gets fed into photosystem 2. And then you keep getting this process cooking along as long as there's light. So you see light has to be striking these two photosystems for all of this to work and flow continually through the day. So in the dark, they can't do this. They don't get that excitation of the chlorophyll, where the electron gets excited and handed off. Okay, so this is a pretty thorough diagram. It's actually showing pretty much all the players in the game here, but we do need to get into this in a little bit more detail. So one of the things that when they pieced this all together and they realized that, oh, there's actually two photosystems that are involved and they're linked in this long chain with lots of other players that are doing the job of passing electrons from donor to acceptor. So again, this idea that ultimately you do get the source of electrons for the light reactions, it's ultimately water. That's where the plant is getting its pool of electrons from. And then the light absorption kicks off the whole process. You'll get electrons getting stripped off of water, given to the chlorophyll in the reaction center. Because if you think about it, when we go back to this image, once light energy comes into the reaction center and that chlorophyll molecule's electron gets excited and off into the triplet state, if that gets donated off, now it's missing an electron. That chlorophyll pigment just donated off an electron, and now it's short one electron. And so it wants to get back into the stable paired electron state. The way to do that is to get a source of electrons from somewhere so that the chlorophyll can get filled up again, so to speak. And that's what's happening here. So as that chlorophyll in photosystem 2, as that special chlorophyll its electron gets excited and then handed off, it's missing an electron. And so here they're trying to show that water will get stripped of electrons to get the chlorophyll back so that it has its full complement of electrons. And you have to imagine that just happening over and over and over. Light strikes into the photosystem 2, electron gets excited, given off, water gets stripped of electrons, giving that to the chlorophyll in the reaction center. So this electron transport chain from photosystem 2, and then you have several players between photosystem 2 and photosystem 1, but eventually you'll have an electron coming to photosystem 1. So now the trick is that chlorophyll will only take on that electron if it's missing one. So we got to get that to happen. So the way to do that, of course, light energy comes into photosystem 1, gets that electron excited and boosted off. So now it's missing an electron. So now it can accept one that's coming from the chain originating back by photosystem 2. So you have to have this light energy hitting both photosystems so that those electrons get excited and boosted off, so you can keep this flow of electrons going all day long. And as long as you can get that to happen, and then the other trick is these two photosystems have to be getting hit with the same intensity and quality of light so that everything works in concert. You want that, photosystem 2 and photosystem 1 to be working together and not out of step with each other. And if all of that happens correctly, then you have this constant electron flow, ultimately from water all the way up and over, so to speak, to NADP+. Okay, so those are some of the big picture ideas on what's going on with these light reaction process. So those photosystems, when they started looking more carefully, by the way, that the fact that they're named photosystem 1 and 2, that's just the order that they were discovered in. That's why they gave them those names. So we abbreviate down PS1, PS2, and they are operating in a series or a chain. And basically what we see when you look at the whole thing from start to finish, light energy has to be used. You get that light energy, now that has enough energy to extract electrons from water that hates to give up electrons. And you end up raising that energy level to get a very strong reductant. So NADPH is a really good molecule. And if you look back, let's go to this one. If you look back here and you see where NADPH is on this scale, you can see it's in the negative part of the scale. So if it's in the negative part, that means it's happy to give off electrons. It's designed that way. And so when the plant makes NADPH, we call that a reducing molecule because now it can go to some other reaction nearby and say, hey, I got some electrons I'm willing to give up to get a certain reaction to completion. Questions on kind of the big picture idea here? Okay. So these photosystems, when you look at them, there is that special chlorophyll I talked about, but there's other stuff. There's some different proteins in there. There's actually several chlorophyll pigments and there's carotenoid pigments. So it's complicated. And then surrounding that reaction center is the antenna pigments as well that are picking up different qualities of light. And then there's a special portion of the photosystem that we call the reaction center. And the reaction center, that's where that very special chlorophyll is that will get excited by the light energy, have its electron excited to a higher state, and hopefully give it off to the right molecule. So they call it the reaction center because they know that's the spot where that particular chlorophyll gets excited and the electron gets given off. So that's the idea that you have light energy being converted. As soon as you start talking about electrons being given off, then you start thinking about biochemical energy. So when you look at the chloroplast and you zoom in to the thylakoid inside, there are, it's kind of weird how things are arranged. You have photosystem 1 and 2 peppered throughout the thylakoid membrane. You have another big player called cytochrome B6F. This is another really big complex. And then there's another, well, this big ATP synthase. So these are all large, big complexes. They span the width of the thylakoid membrane. They're big and bulky. And so we call them integral membrane proteins, or I like to say transmembrane because it's shorter, meaning they span the width of the thylakoid membrane. And when you look at how they are stuck into the membrane, they actually say it's a vectorial arrangement, which basically that just means that it's the way it's stuck into the membrane and what components are the arrangement of that through the membrane. It allows for certain reactions to occur on either side of the thylakoid membrane. And so it's well designed. And that will make more sense as we work through this a little bit more. But basically, this vectorial arrangement allows for water to get stripped of electrons in one part, and then NADP plus get reduced in a separate part. And so basically, they're occurring on opposite sides of the membrane. And that's actually important. And I'll explain, you'll see why as we work through this. So when water is getting stripped of electrons, water is located in the lumen portion of the thylakoid. So remember, if you crack open the thylakoid and look inside of it, We call that the lumen area, just like your intestines. If you were to peer inside your intestines, that would be the lumen of your intestines. And then NADPH and ATP are all being produced in the stroma of the chloroplast, which that's just the watery portion that the thylakoid is floating around in. And so this setup is really, really important in order to get really good flow of electrons. And then you also end up with production of ATP and NADPH actually right where it's needed, because when it's in the stroma getting produced there, that's actually right where it's needed to do the fixation of CO2. So it's nice and handy that way. It doesn't have to travel any distance to do its job once they get made. Okay, so those, the photosystem 2, when you look at the thylakoid, it's weird. You see a lot of photosystem 2 peppered through the grana region. So that, remember, the grana region is the stacked region of the thylakoid. You see lots of photosystem 2 showing up there. On the other hand, you see photosystem 1 and the ATP synthases, they seem to pepper and populate the stroma lamellae area, which is relatively unstacked region. So it's kind of clustered. You have these clustering going on. It's kind of weird. And then that another big transmembrane is the cytochrome B6F. That one, when they study it, it looks like it's pretty even, Steven. It's pretty evenly dispersed throughout the thylakoid. So what that means is, in order to get all the electrons to flow nicely from start to finish, and to get things cooking along so you can get some NADPH and ATP made, if they're all clustered kind of away from each other, and yet they have to link to each other, you have to have some other carriers involved to kind of move around and get electrons to where they're needed. So we do see in this process, there'll be some so-called mobile electron carriers that can shuttle electrons between these different big complexes that don't move. All right, so once these get made and put into the thylakoid membrane, they're kind of stuck. They just stay there in their physical location. And yet they have to get their electrons passed through this nice chain of donors and acceptors. So the mobile carriers are going to do that. They're going to help get the electrons to where they're needed. So if you look at a thylakoid membrane, here they're just showing a section of it, and they're kind of zooming in on mostly the grana in this area here. This is the grana. And then the stroma lamellae they have going off to the sides here. But if you look at the color coding, you can see that there's a lot of the olive green as photosystem 2. So you see a lot of that peppering into the grana region, whereas the photosystem 1 is more the lime green, and you see that showing up more in the stroma. So you have this clustering of these big complexes, and they don't move. Once they're there, they stay put. And so that is why you need to have these mobile carriers to get the electrons to where they're needed. So when they map it out and they look at it more carefully, here's actually results of years upon years of work looking at the complexes of these transmembrane proteins and, big complexes. So photosystem 2 over here. So if you've taken Intro to Cell and Molecular and you've seen designs of proteins with the alpha helices and beta sheets, that's what's being represented here. And it's really pretty. You got a lot of different subunits, different players that make up these transmembrane proteins. And so you see photosystem 2 over here and photosystem 1. And then they're showing electron flow between them. You have another big complex in between called cytochrome B6F. And then we can't forget about, of course, the ATPase. So this effort to try to figure out what makes up photosystem 2 and photosystem 1, like I said, decades of work going into this. And you can see just by the different colors and the different shapes of the proteins, there's a lot of complexity to these big transmembrane complexes. And then here you see this little PQH2. That's a mobile carrier. Very simple, very small, very mobile. So we'll start talking about all the different players. Let me go back to this picture. So here, the other thing on this image, they're showing LHC1, LHC2. So that stands for Light Harvesting Complex, Light Harvesting Complex. So that's their fancy way of talking about antenna systems. So the antenna systems that surround photosystem 1 are called Light Harvesting Complex 1 molecules. So that's where you see all of the green here is surrounded by the purple. All right, so they're trying to show that those LHC1s are surrounding. I'm sorry, LHC2, the light purple, is surrounding the olive green photosystem 2. All right, so they're trying to show that there, that you have those antenna pigments or LHC systems. And then with photosystem 1, more the lime green, you see the dark green LHC1. All right, so those, again, those are the antenna complexes that are surrounding photosystem 1. So depending on what species you're looking at, what organism that's doing photosynthesis, you'll see some differences in these antenna systems. That's why they give them different acronyms. They don't just say antenna system. They call those, like the ones we just looked at, LHC1, LHC2. But no matter what variation you see there are still pigments that are absorbing light energy. That's their job. And their job then is to transfer the energy, that light energy, from pigment to pigment to pigment and get it into the reaction center that it's associated with. And so we often say it's funneled, that light energy is funneled to the reaction center. And it's in the reaction center of those photosystems where you see that special chlorophyll that will then get excited. Its electron will get excited. So they are hugging around the photosystems, those antenna systems. They kind of surround and interact closely with the photosystems. And the ones, the LHC2 and 1 that they've been studying quite heavily, they say there's like 15 chlorophyll A and B in angiosperms. But they still are investigating all these different antenna complexes and pigments, what the ratios are. It depends on the organism and the species. The other thing that seems to be part of any antenna system that you look at is carotenoids. Those pigments seem universal. And so, at least as far as I know, they have yet to discover a photosynthetic organism that does not have carotenoids. It just seems incredibly universal, whether you are a bacteria doing photosynthesis or an angiosperm plant. So carotenoids are very, very important as helping with the absorption of light energy, and they play other roles as well that we'll talk about. So again, going to the antenna system idea, here they're saying, yep, there's chlorophyll A and B and carotenoids, and all of them are surrounding the photosystems. They're absorbing light energy, funneling that light energy through inductive resonance to the reaction center. So we've seen this before. So once you get that energy into the reaction center, they're trying to show that that really gets some nice excitation. And so that's why they've drawn it further up the energy scale here. They're trying to show that it got excited, the electron got excited with that energy. So here is just, they've zoomed in on the light harvesting complex that associates with photosystem 2. And again, you can see with all the beautiful colors and designs here, a lot of subunits and different proteins and cofactors involved. You can also see how it's spanning this kind of purpley gray rectangle. That's the thylakoid membrane. And so they're showing how it's spanning, these light harvesting pigments also span the thylakoid membrane. So in simplified form, you could represent it like this. So what we're looking at here is a stretch of the thylakoid membrane. And the lumen portion, so that's the portion that's in, the internal part of the thylakoid membrane. And then if you look up high here, the stroma is the watery portion that the thylakoid is floating around in. Right, so you have your thylakoid, and then you have your transmembrane proteins that are involved in the electron transport chain here. So we're going to use this a lot to talk about what's happening, but this isn't showing all the players of the light reactions, but it's a nice kind of simplified way of looking at it to get our bearings on this process. So they're using black arrows, black dotted lines for electron flow. And then they're using the blue swoopy lines here for proton movement. But what you see here is they are showing water when it associates with photosystem 2. We're going to see that water can get stripped of electrons. So they're trying to show that idea. And then the electron flow is the dotted line, the black dotted line. You see that light energy is hitting both photosystems. Hopefully it's doing it just right so everything stays in step with each other. So we see photosystem 2 and photosystem 1. We also see cytochrome B6F hanging out, and then the ATPase. So we like to draw it this way in order to talk about the process of photosynthesis. But I'd like you to appreciate that that's not how it's lined up in the thylakoid, which is kind of weird, but it's clustered. All right, so you don't have this beautiful line of molecules like this. It's just, this is our way of easily talking about it, but you have to remember it's a little messier than that when you actually look at the thylakoid. Okay, so just keep that in mind. And then this figure, I think, is from this, well, it's from a different book. So either one, it's basically showing the same thing. One fits better with your brain than the other. Okay. So normally, when we talk about the light reactions, we tend to start thinking about that process at photosystem 2. What's going on with photosystem 2? So that's what we'll do. So if you can imagine light energy getting funneled into the reaction center of photosystem 2, there's a couple of things that are going to happen. You're going to get the chlorophyll, CHL, that special chlorophyll pigment, one of its electrons is going to get excited. So the way to depict that is if you write P680, you're implying you're talking about the reaction center chlorophyll, that special chlorophyll. And if you write P680 asterisk, now you're saying its electron got excited. All right, so that's how we denote that. Whoops. And so we're going back to this idea. So this is the special chlorophyll in the reaction center. It got hit with light energy. Its electron got boosted to a higher energy state, hopefully to the triplet state where it can stay around long enough to get passed off to a donor. So now we have that excited electron. Now we have to get it passed off. So then you would say, well, okay, the excited chlorophyll, P680 asterisk, will donate that electron off to the correct acceptor molecule, which is pheo. Which stands for pheophytin. Pheophytin. It's a modified chlorophyll molecule, if you're curious. When we talk about this and write it out or on a quiz or exam, you can shorten everything. Like you don't have to write out pheophytin, you can just put pheo, and that's fine. So now what's happened is that electron got given off by the chlorophyll and got handed over to pheophytin. So now we depict it this way. We say, okay, that chlorophyll in the reaction center, the P680 chlorophyll just lost an electron, so now it's in the plus state. Pheophytin gained an electron, so now we put it in the negative. So that's showing what they call a charge separation. You have a oxidized chlorophyll, reduced pheo, plus minus. So that's the exact spot where you can say light energy has been converted to chemical energy. You had a redox equation, redox process result from that. So this is photosystem 2. And so there's certain regions in here where you're going to have the pheophytin associated with photosystem 2. It's not a part of, it's associated with photosystem 2, let's put it that way. And then, let's see, where's the other image I wanted to show? Is that? Okay, so let me go back to this one. So here, this is photosystem 2. One of the things that they've labeled is OEC, and they put it, it's right in this area here. So that's part of photosystem 2. That's the oxygen evolving complex. This is a mock-up in better detail of what that oxygen evolving complex looks like. There's been intense study of the oxygen evolving complex because the idea that plants can split water, can extract electrons off of water is pretty phenomenal. That is such a hard thing to do. And so they have been intently looking at the oxygen evolving complex. And when you look at its operation, basically, yes, it is through the help of light energy coming in to photosystem 2. It provides a mechanism where the oxygen evolving complex can strip electrons off of water. And so basically they tend to show it this way with this type of equation. 2 waters plus yield oxygen, 4 protons, 4 electrons. At the same time, what you have happening is as soon as that electron is stripped off of water, it's given to that chlorophyll that's missing an electron. So that P680 plus grabs that electron and now it's fully reduced. It's chemically happy and stable, and it's ready to get hit with light energy again. And then these protons that are being shown as part of the products, those we tend to say they get dumped into the lumen portion of the thylakoid. So that's being shown here on the diagram, how you have water getting stripped of electrons. And then here, if you go back to this image, they're showing it here in the lumen area. That's where you have the protons getting dumped after you get electrons stripped off of water. So that's really good because this buildup of protons in the lumen, if this keeps happening over and over and over and over, you get a lot of protons coming off of that reaction pathway. That's good because now you start to get protons building up on one side of the membrane compared to the other. And so then once you start seeing that, that's a proton gradient developing across a membrane. And that's great because that sets up that's potential energy that can be used. And in this case, we see it's actually being used to allow for ADP and phosphate to interact and get you that wonderful ATP. So anytime you see a proton gradient getting established across a membrane, whether it's the thylakoid, the mitochondria, a cell membrane, whatever, proton gradients are awesome. If organisms can manufacture and design a way to get that to happen, one of the great things that can happen as a result is ATP production. It can do other things with a proton gradient. It's not always ATP production, but that's one of their favorite things to do. And you're doing that right now in your mitochondria. That's what's happening, hopefully. So, okay. All right, so let's go back. All right, so photosystem 2. So now I just told you that electron goes to pheophyton, but you don't see any pheophyton here. It's not labeled. So that's what I mean by this diagram is not detailed. It's more of a big picture idea. If you go back to this one though, where is it? No, where is it? I lost it. It's here somewhere. If you go back to this one, what we just described is here at photosystem 2, we described how light energy excites that chlorophyll electron, and then the electron gets given off. Here's pheophyton. Okay. So pheophyton is the first acceptor, and it's associated with photosystem 2, kind of bound loosely to photosystem 2. And then we also talked about how water gets stripped of electrons in photosystem 2 by that very special area called the oxygen evolving complex. And so it's kind of interesting they named it that. I guess from a human perspective, we care that there's oxygen being evolved, but from the plant's perspective, they're like, yes, okay, here's some oxygen, but I don't really care about that. I'm going for getting my electron flow going. I like the protons building up. Oxygen, the plant's not too concerned about that. Okay. So now pheophyton, you have to imagine pheophyton here. It's going to do its job, which is, so pheophyton is right here, going to do its job of donating the electron to the next acceptor in the line. And that is going to be the PQ. So that stands for plastoquinone. But again, you can abbreviate down to PQ. So plastoquinones are small molecules, and they are there right associated with photosystem 2 and next to pheophyton. They're there. And the plastoquinones, when you look at them chemically, they're what we refer to as a 2-electron gate. And what that means is plastoquinones, in order to be chemically stable and happy, so to speak, they need 2 electrons, not just 1. So when they are going through this process of accepting an electron from pheophyton, they'll be like, okay, yeah, I got 1, but I need 1 more. All right. So just, I tend to think of plastoquinones are kind of a hog for electrons. They're greedy. They want more than 1. And so before they can get out of the gate, I often think about, horse racing, how you have to get out of the gate to start the race. You need 2 electrons in order to get out of the gate and do your part. So what's going to happen is pheophyton will get that electron from the chlorophyll, turn around and pass it to a plastoquinone. There's actually a couple of plastoquinones involved in this process. So we say it goes to the first PQ. The first PQ turns around and gives it immediately to a second PQ sitting right there. So now that second PQ has 1 electron, and not good enough. It's like, I need 1 more. And so what happens is everything repeats. Now, when I say everything repeats, I mean all the way back to light energy coming into photosystem 2, exciting that chlorophyll, getting that electron boosted, hand it off to pheophyton. Pheophyton gives it to the first PQ again. First PQ gives it to the second PQ again. So all of that has to happen all the way back to light energy striking photosystem 2. Once that happens twice, essentially, so that second PQ has 2 electrons, then it's almost done. But in order to be fully chemically stable, it has to grab 2 protons. So it's going to pick up 2 protons from the stroma. That's important that you know where they're coming from. And then once it's had its 2 electrons that it's received, it grabs 2 protons. Then we refer to it as PQH2. That's its fully reduced form. That's its chemically stable form. And now it's able to do its role in the light reactions. So now it's got these electrons. We're going to see, they're trying to show in this diagram that it moves between these two complexes. So that's its job. It has to go and deliver electrons to cytochrome B6F. Okay, that's a lot being thrown at you today. And so please spend some time looking this over before next lecture. We'll go through this again a little quicker, and then I'll build on it and show the next half of the process. So have a good weekend.